Good afternoon, everyone. My name is Susan Moyer, and I've had the pleasure today of hosting Dr. Max Cooper. And welcome to the Wednesday afternoon lecture series. And today we have also a guest for the introduction. I'd like to introduce my boss, Dr. Anthony Fauci, the director of NIAID, who will be giving the introductory remarks today. Thank you and enjoy.
Thank you very much, Susan. It's really a pleasure for me to be here today to introduce Max. Good afternoon to all of you, and welcome to the NIH Director's Wednesday afternoon lecture series. Max Cooper will be speaking to us about his insights into the evolution of adaptive immunity. I want to tell you very briefly a little bit about Max and a little bit about his accomplishments. First, a quick thumbnail about his background. Many of you in the audience know him, but I think more people have just heard of him. Max was born in rural Mississippi and graduated from the University of Mississippi, and he earned his MD degree and completed his residency training in pediatrics at Tulane. He conducted postgraduate research at the Hospital for Sick Children in London and then became a trainee in allergy and immunology at UCSF. And along the way, he developed a special interest in clinical immunology from his experience caring for children who were unusually vulnerable to infections. He became an instructor in pediatrics at Tulane and then moved to the University of Minnesota before settling down the roots of his academic career at the University of Alabama in Birmingham, beginning in 1967. After spending decades there, in 2008, he moved to his current position as professor in the Department of Pathology and Laboratory Medicine at Emory University School of Medicine. Now a bit about his accomplishments. Could I have the slide, please? By the estimation of virtually every knowledgeable immunologist who has a corporate memory, Max Cooper is clearly one of the true iconic giants in our field. His early work was crisp, eloquent, and transformative in the field of immunology, and to this day he continues to lead us. 50 years ago at the University of Minnesota, Bob Good, Max, Ray Peterson, and their colleagues published a seminal paper in Nature that is going to be celebrated here as shown on this slide that began Max's remarkable string of research accomplishments. His experiments with chickens reported on in that paper began the elucidation of separate T and B cell lineages and the identification of hematopoietic tissue as the origin of B lineage cells. Bridging these insights to the clinical arena, he and his colleagues have for decades been performing comparative studies of lymphocyte development and function with an aim to better understand the cell differentiation defects in individuals with immunodeficiencies, lymphoid malignancies, and autoimmunity. Max is a truly very modest person, and I'm sure he's going to be upset with me for saying this, but virtually anything that any of us do in the fields of immunology, autoimmunity, hematologic malignancies, immunodeficiencies, has been impacted in one way or another by Max's seminal work originating back decades ago, but continued to this day. However, seminal research accomplishments are not all that Max is about. He has been recognized for his mentorship skills, serving as a valued advisor for students, postdocs, and junior faculty. And one of the reasons that I'm introducing Max is a little bit of history. I first met Max around 1970 when I was in the middle of my fellowship here at NIH. I was presenting a poster at an annual meeting of the American Association of Immunologists, and at that time, no one but my family knew who I was. Max was already quite a famous individual, and I had only heard about him. He came over to my poster and spent a considerable amount of time chatting with me and making constructive suggestions. I was awestruck, but he treated me like a colleague. I was totally ecstatic, and over the years, to my great benefit, we have become very close friends. I have used that experience as my standard of always treating young scientists and trainees as colleagues. They will never forget it. I certainly did not forget that first day with Max Cooper. Max is a member of a number of prestigious and honorific societies, including, among others, the National Academy of Sciences and its Institute of Medicine, the American Academy of Arts and Sciences, the ASCI, the Association of American Physicians, Fellow in the Royal Society of Medicine, former President of the American Association of Immunologists. He's won many, many awards, including the 3M Life Science Award, the Sandoz Prize for Immunology, the Avery Landsteiner Prize, the Robert Koch Prize, and on and on and on. His most recent studies, which he will discuss today, have focused on a search for the origin of our adaptive immune system and have revealed that the jawed vertebrates and jawless vertebrates, lampreys, and hagfish use different strategies for generating large repertoires of lymphocyte receptors for antigens. So please join me in warmly welcoming Max Cooper to deliver the NIH Director's WALS lecture.
Tony, thank you for that very warm and generous introduction. As you can see, I've been around for a long time, and I'm a bit of a dilettante in research in this field. I'd like also to thank Susan for her kind hospitality, and Francis, thank you very much for the honor of presenting the NIH Director's lecture today. Now, as all of you know, every living thing on our planet in this struggle for survival has an immune system of some sort. Even an adaptive immune system, as we know from recent studies of the CRISPR-Cas system in bacteria and eukarya. And we're all quite familiar with the toll-like receptors that were elucidated best first in insects. But it's only in our vertebrate lineage that we have a lymphocyte-based adaptive immune system with a huge repertoire of specific receptors that will allow recognition of almost any configuration, pathogen, and respond with and produce memory. And we being Jan Klein and I, just after the turn of this century, decided to join forces to go back and see if we could trace the roots of our adaptive immune system. We knew that all jawed vertebrates, sharks and onward, had the same basic thymus, where T-cell receptor genes are rearranged under the influence of RAG and B cells with antibody genes, VD and J rearrangement, and class 1, class 2 to present antigens, peptides to T cells. We would have liked to go back to some of the more primitive or earlier jawed vertebrates. But unfortunately, the only two living vertebrate members that are considered to be more basal than jawed vertebrates are these two lamprey and hagfish, jawless vertebrates. Now, we chose to start with the studies of lamprey to start with, but as Al mentioned, we also have studied hagfish. And we've studied, there are many different kinds of lampreys, and they are worldwide in distribution. One of the more than half of which are predatory is the sea lamprey, and we've studied them mainly in their larval phase, where they spend most of their life before becoming, undergoing metamorphosis and becoming predatory, mating and then dying and starting the circle over again. We studied them mainly at a time when they're 3 or 4 years old, and if you cut one of those larvae in half at that age, one of the initial findings that started us on this study was Jan Klein's group had found a transcription factor that's important in hematopoiesis, and it seems to be particularly important in B lymphopoiesis, as Tony mentioned, a long-term interest of ours. And he found that a cell in this hematopoietic tissue called the typhlosoil, the simple intestine is wrapped around it, was expressing a SPI-B transcript, a member of the PU.1 family. And the cell was round, and it could have been a lymphocyte. So Jan and I decided to join forces and to look for the roots of our adaptive immune system. We had several questions in mind, some specific, some fairly broad. For example, I was curious about which came first, B cells or T cells. We also wanted to know how an adaptive immune system blended in with a pre-existing innate immune system. And we thought if we could get back closer to the time of that overlaying, we might gain more insight. Our strategy was to just take cells from this typhlosoil area and by simple 90 degrees, 180 degree light scatter, pick out cells that looked like lymphocytes with regular blood stains, right Gamza, and even by EM had this very simple morphology of a lymphocyte, which is, of course, very nondescript. So we took these cells and made cDNA libraries and got what, at that time, was high-throughput screening. We found many of the genes that our lymphocytes use for activation, migration, and so forth. What we didn't find were any of the cardinal elements of our adaptive immune system, which was disappointing, especially to me, but we went back and stimulated these larvae lamprey with a cocktail of live bacteria, plant mitogens, and foreign erythrocytes, and then made a subtractive library, and Zev Panzer, who had joined our lab then, made this subtractive library, and we found that it was enriched with leucine-rich repeats, which wasn't what we were looking for. But a summer student sequenced the first 22 open reading frames, or almost completely open reading frames, to show some very striking features that made us recognize that this might be what we were looking for, although we hadn't realized that. These were the feature that struck one immediately is that every one of them had a different sequence. They differed in the number of these leucine-rich repeat modular units, and even the ones that they shared had very different sequences, one from the other. Now, the longest one, here's a stick model of it, and it shows that like many leucine-rich repeat proteins, and I should mention that every living thing on our planet, as far as I can recognize, uses leucine-rich repeats for various purposes. These had, all of them had an N-terminal and C-terminal so-called capping LRR, which had been shown to protect these kinds of proteins, the solenoid structure, their core hydrophobic sequences. And they all had an LRR1 of about 18 residues and a connecting peptide of 13. In between, these green blocks were the ones that were variable in number. They were invariably 24 residues. The stalk region was kind of interesting too. It was rich in threonines and prolines, and it had 8 cysteines in this terminal, carboxy terminal region, the hydrophobic sequence here. In addition, though, there was a predicted GPI cleavage site. And when Gertz Ehrhard expressed a construct for this with a Ha tag in mouse thymocytes, he found that it was on the surface and it could be cleaved from the surface by the enzymatic action of lipo enzyme that would cleave GPI linked proteins of bacterial origin. Now, when we sequence more of these from many different larvae, and both adults as well as the larvae, the sequence is depicted on this cartwheel phylogenetic map. And you can see that for each one is paired with its closest relative in terms of sequence similarity. So this one is about 10, 20-something residues different from the other one, which is it's closely related to. And all of them were different except for 3 pairs, which came from these immunostimulated larvae that I mentioned earlier. There's another feature shown here on this slide. The asterisks indicate single-cell PCRs, and in each instance, only one sequence was found, suggesting monoallelic assembly or expression. Now, when we looked with Chris Amamiya in Washington, who had a library, genomic library made from the sperm of a single lamprey, and used our primers to screen this library, cDNA library that he had, a back library, we could only find one VLR gene, not multiple ones that could account for those different products that we had been seeing, and it was very incomplete. It only encoded for part of the N-terminal LRR and parts of the C-terminal LRR, the stalk region, and a signal peptide. In between, there were large stretches of about 6 kb each of non-coding intervening sequence. But when we got sequences, genomic sequences of flanking regions, we found that there were LRR coding cassettes, if you will, that if somehow stitched in to this germline VLR gene with deletion of the intervening sequences could account for the kinds of mature VLR sequences that we had been seeing. And indeed, when we looked at isolated lymphocytes and erythrocytes only made with forward and reverse primers, this product representing the germline, we call it variable lymphocyte receptor gene. Whereas in lymphocytes, we also found smaller ones, and when these were cloned and sequenced, all of them were different, just as the ones that we had been seeing. Now, we don't understand all of the biochemical basis of this assembly process, but this slide illustrates some of the things that we know about it presently. These flanking sequences are brought in, and based on short stretches of sequence homology between the donor and the recipient portion of the incomplete germline gene, these sequences get copied in to the gene and replacing the intervening non-coding sequences at the same time. That process occurs over and over in a stepwise and piecewise fashion until there's complete assembly into a mature VLR gene. The process, as Nagawa and his colleagues could show, can start on either end, but not on both, and go through to completion. Now, all of these pieces, and we didn't recognize it so well to begin with, or only bits of them are used because they're stop codons and so forth. This process, how the DNA gets cut and the donor sequences are used as templates to be copied in bit by bit and step by step, is thought to be catalyzed by 2 AID-like gene products that were identified by Igor Rogozin and Zev Panzer and their colleagues. One of them looks very much like our AID gene that, as you know, is involved in class switching, somatic hypermutation, and also in gene conversion that's used by some species, chickens and rabbits, for example. The other one is intronless. This one is more like this. Another one that they call cytosine deaminase 1, this one too, is intronless and could have gotten into the genome potentially by retrotranscription. I'll come back to those two CDA1 and 2 again in a moment. But at that time, the model suggested that these must be assembled and expressed on lymphocyte-like cells. And Igor Rogozin and Lax E estimated from sequences that we provided them that the repertoire of these would be greater than 10 to the 14th. At least the same general repertoire that our B cells generated by light chain and heavy chain VDJ rearrangements. And just to see if those were actually capable of being made into antibodies, we repeated an experiment that had been done back in the 60s by a couple of groups, whereby these lamprey larvae, and you can see they're pretty small, here are the fingers of the person injecting with this insulin syringe. So you inject a small amount. In this case, the antigen was erythrocytes and human O-positive erythrocytes. And as had been reported earlier, that resulted in a couple of weeks in all of the animals making agglutinins of a fairly low titer. But if we boosted them, the titer was increased by about 20-fold. Now, these agglutinins, the biochemistry of which had evaded investigators, we thought might be the VLRs, and we made a monoclonal antibody to the stalk region of our VLR and used it to adsorb, and all of this antibody activity was removed. Now, the cells that were producing these agglutinins, these variable lymphocyte receptors that were secreted, are shown here in these stained green in the sections of gill folds, these being the filaments. And before and after immunization with anthrax exosporium. And these have also been injected with BRDU. And so these yellow cells, you can see more cells that are dividing after immunization than before, suggesting that these respond to antigen stimulation with proliferation. Now, the cells that are actually secreting the agglutinins, or the VLRB antibodies as we call them, are being made by cells that are even like plasma blast, as shown here, or even mature plasma cells. Brant Heron, who's done a lot of the work to define these Lamprey antibodies, used this anti-stalk region antibody to show that a Western blot of the serum from these are very large molecules, variable in size, as you might expect, because they have different numbers of these LRR blocks, but treatment with 6-mercaptopurine to reduce the disulfide bonds dropped them into smaller units, again still variable. Now, in order to study the structure of these, Brant devised a way to make recombinant monoclonal VLRB antibodies by immunizing, making cDNA libraries, expressing them clone by clone in human embryonic kidney cells, and then picking out ones that secreted the VLR that could bind his antigen. And looking at a pure VLRB antibody, we could show by negative EM that they look much like an IgM antibody molecule, except they're only made of one kind of chain. They are bonded at their base, the stalk region, by those cysteines that I mentioned earlier, and at least 4 of the 8 are required for this binding and configuration. So these are multimeric units with high avidity, even when the individual's reactivities have a low affinity for the antigen. The one that was specific for this H trisaccharide, the human O-positive blood group determinant, Byung-Woo Han and Ian Wilson's laboratory was able to obtain crystals and show the structure. Here's the H trisaccharide antigen. These are hydrogen bonds, 1 to LRRV2, 2 in the N LRRV, and I haven't mentioned, but there's a highly variable insert in this carboxy LRR, and it forms a loop, and it too figures strongly in hydrophobic bonds to this H trisaccharide. Now that looks more like this in a space-filling molecule. And Roy Mariuzza and his colleagues working with Zev Panzer have also shown for protein antigens, and so have we, to a predominant coat protein of the anthrax exosporium. But the point of specificity that's shown in this slide, this is that RBC36 recombinant monoclonal, and it reacts with everything that has the 2 terminal sugars of this trisaccharide in the proper configuration. Fucose with a 1,2 linkage to the galactose with a 1,4 linkage to the next carbohydrate. This is a glycomics center that Rick Cummings at Emory and his colleagues have designed. It has 611 different carbohydrate moieties on here. Every one that it reacts with, and it reacts with nothing else, has these last 2 terminal to show that specificity. So at that point, it looked as if these incomplete genes were put together in the fashion that I've just alluded to, and the cells responded to antigen to proliferate, differentiate, and secrete these antibodies. So I thought that proved my bias that B cells came first. However, when we had looked in hagfish, we had found 2 VLR genes. Then Rogozin and Panzer and their colleagues found a second Lamprey VLR gene, and most recently, Kasahara and his colleagues have found a 3rd VLR gene. They all are incomplete, even some more incomplete than the original VLRB gene. And they also, not shown here, are flanked by donor LRR sequences. When Pongoa and Masahirano made the stalk regions are invariant for each one of these, and they differ from each other. So monoclonal antibodies were made that discriminated between VLRA, B, and C, and those were used to show that each one of them is expressed by an individual population of lymphocyte. So the VLRBs are different from the A's, C's different from the A's, C's and B's also separate populations. That allowed us to enumerate these cells, isolate them, and begin to look at their functional properties. One of the striking features is that the VLRAs in red and the C's in blue preferentially respond to injection of a classic T-cell mitogen in jawed vertebrates, PHA. Whereas the VLRB cells that I've been talking about up till now respond very little. Moreover, when we looked at a limited gene expression profile for the A's, B's, and C's, as you can see, many genes were selectively or preferentially expressed by the VLRAs and C's together. Note to start with that the CDA1, this cytidine deaminase 1, is preferentially expressed by A and C, not by B, and CDA2 is preferentially expressed by the VLRBs, implying that perhaps they are differentially involved in the assemblies of these different types of variable lymphocyte receptors. The VLRAs also have a number of genes that are transcription factors, chemotactic factors, and signaling elements that are used for T cell development and function on this scale, and I won't read all of them off. In addition to some cytokines like an interleukin-17 D-like and migration inhibitory factor, whereas the VLRBs express transcription factors and other signaling components and elements that are characteristic for mammalian B cells. I'll come back to these differences in a moment. So one of the questions that immediately we wish to answer is, where are these genes assembled? And in other words, where is a thymus equivalent-like tissue for these T cell-like cells that respond preferentially to PHA? They also don't secrete their products. They also, and I won't show the data, preferentially respond with proliferation to allo stimulation. So we looked at the most likely place, turned out to be in the gill region, and there were several clues that this might be the case. With Thomas Boone and his colleagues, using in situ hybridization, we could show that FOXN1, a gene that's essential for stromal cell development of the thymus, and the mutated gene in nude mice and humans as well, was only expressed at a level that could be detected in the tips of these gill folds and at the ends of the first filament. This cytidine deaminase A1 was also only detectable in that site, and even if you stimulate it with PHA or immunized in other ways, this is the only place that this enzyme thought perhaps or implied to be involved in the assembly of the VLRAs and the Cs is expressed in this location. VLRA is also expressed here. VLRB is not. It's not a very impressive tissue, but there are lymphocytes in here in close juxtaposition with epithelial cells in this environment, and we called it the thymoid. Each one of the tips of these gill filaments express this CD1, and a few of the cells in this thymoid region expressed A but not B. They also can express the C protein. When Thomas used microdissection to get samples of DNA from this thymoid region, he found that there were lots of non-productive assemblies of VLRA, and these were not found in blood, which was consistent with the possibility that the assembly was being selected for in this location. For the Cs as well, the same story, except that there were also non-functional assemblies in the blood as well as in the thymoid, although they were far more common in the thymoid region. Now, many of these were incomplete, and they were particularly informative in that you could see how the VLRC in this instance is bringing in these donor LRR sequences, first this, then adding more for the LRR1 to complete it, an LRRV, and then more. And it can begin, these incomplete assemblies involve assembly from either 5' or 3' locations. I show this VLRC because it's a simpler donor sequence. There are only 5 different kinds of potential donors, and you can see how they would have to come in a sequential manner for expression. There are over 180 of them, and I won't, for time reasons, go into more. When Masa Hirano pulled together cells from all of the different tissues and discriminated between the germline and mature VLRA, C, and B genes, he obtained several bits of information. I'll point out just a couple of points. One is that the VLRA-B positive cells only assemble and express VLRB transcripts. The VLRA and C loci are silent. And similarly, the A positive cells are the B, VLRB is totally silent, and the same thing is true for C, but there's some shared assembly of A and C that are both, as I've shown you, evidence suggesting that they are assembled in this thymoid region. Our data suggests this model, working model, that a cell of a common progenitor, thymocyte, begins assembly on one allele or the other. If it's productive, then the cell can survive. If it's non-productive, it can try again on the other allele. If it misses on getting a productive assembly on both alleles, it still can can assemble a VLRA for survival. The VLRAs seem to begin assembly later, and they go through the same alternative possibilities. Now, the VLRAs and Cs, which are beginning to look more and more like our T cells, they migrate preferentially into endothelial surfaces. This is the typhosol, this is the intestine, and you can see these green and red ones here in the intestine mixed together. There are no Bs not shown here. And in the skin, these VLRCs are preferentially expressed by themselves. And moreover, in the skin, those VLRCs are oligoclonal, whereas they are not very oligoclonal in other tissues, and VLRAs are not oligoclonal in any of these sites. So it's beginning to look more and more like alpha, beta, and gamma delta T cells. And now let me come back to these differentially expressed genes. SOX13, which is an inhibitory transcription factor, has been shown to interact with TCF1 to inhibit, which is essential for T cell differentiation in mice. A TLR3, an interleukin-16, and also integrins that genes that encode integrins are homing molecules for migration to epithelium. So it looks as if there's a striking resemblance of VLRCs to gamma deltas, although not exactly the same, and you would not expect them to be for a time of difference since their emergence in evolution. And the bees, of course, much like the bees, bee cells that we have. So what about the hagfish? And hagfish turn out to have also 3 VLRs that we've named VLRA, C, and B. And these are expressed preferentially by 3 different lymphocyte lineages. Their products, the lamprey and the hagfish, VLRA, C, and Bs, are very similar. The predicted structure and actual structure of these are similar. They even have the same preferential selection of the number of these insert links, which is short for this VLRC, so that it doesn't have this loop that I mentioned earlier, perhaps a clue to what it's actually seeing. So I won't go into that part of our study, and we've conducted other studies with Roy Mariuzza and his colleagues to gain evidence for suggesting selection in the thymoid region for these VLRCs, but it clearly says that these 3 different lymphocyte lineages the genetic program must have been present in a common ancestor of jawless and jawed vertebrates somewhere around 500 million years ago. But the VLRA system and the B cell and T cell receptor systems that we have, we jawed vertebrates, must have been convergent solutions to a mechanism for generating a highly diverse repertoire. Now, exactly what these receptors might have been seeing before convergent evolution of these two types, we can only guess. But one of the questions that I'd like to ask quickly in the last few minutes is, when did the innate lymphoid cells and natural killer cells evolve? As you know, in the last 5 years, these non-cytotoxic lymphoid cells of different types that some call helper lymphoid cells, none of which express the T or B cell receptors, have become more and more interesting and seem to be involved in defense, particularly along mucous surfaces, initiation of the adaptive responses of T and B cells. And they've been categorized into several different lineages, these ILC lineages for natural killer cells, and 3 different types of ILCs, 1, 2, and 3. And so we wondered if we might be able to find any evidence for when these evolved. So we went back and looked at the cells in the lymphocyte light scatter gate that were negative for all 3 of these different specific VLR types of receptors. And as you can see from this red, in all the tissues that we looked in, blood, kidney, typhosol, and especially in the gill region, was abundance of these triple negative cells. So you may have noticed as I've gone through, these triple negatives are just a blank for any of the orthologs or lookalike genes that I've been talking about that are selectively expressed by A and C and B lineages of cells. But when we looked with some of the marking genes that have been shown to be important in differentiation of ILCs and natural killer cells, PLZ, ID2, infill, KIT, and so forth, you can see that these are preferentially expressed by these triple negative cells. Also a cell surface molecule that can be found on NK cells, and a couple of other genes that we don't know the significance of. So if you look on this map that David Artis and Jürgen Spitz have recently produced for the common lymphoid progenitor and the different transcription factors that are important for differentiation of IL-1, 2, 3 lymphoid tissue inducers, which are essential for developing of lymph nodes and Paris patches, none of which the lampreys have, by the way. And NK cells, the genes orthologs that we can find are suggestive that indeed at least this ILC2 like lineage, the transcriptional program, is awfully reminiscent of that shown to be required for its development or their development in mice. And as also the NK cells with ID2, the CD56, and these are large granular lymphocytes, function of which we've not yet shown. So for an NK-like, and at least this ILC-like too, it's awfully suggestive. So the way we would try to put these findings together is that 2 main points, that T and B cells have been inseparable companions for over 500 million years. The question is, why? And why have they been selected for and selected for further evolution over this long, long period? I would submit that it's probably based on their value in protection against infections. One could also use particularly recent data to suggest that the old idea that the T cells were selected and the immune system evolved for protection against malignancies, that is also a potential tenable reason for the constant evolution of T and B cells for this very long period of time. And the slim data that I showed you at the end may suggest that the basic genetic programs for innate lymphoid cells evolved even earlier than the T and B cells, for which two convergent solutions were obtained to give them specific receptors. These are people whom I mentioned who did the work that I've been talking about. Brant Heron, who's done almost all of the antibody work, Masa Harano for an amazingly large amount of what I've been talking about the separation of the different populations and their genetic programs. Sabi Das is a computational biologist who's done much of the phylogenetic comparisons. Swaling Yu involved in the antibodies that we are analyzing and that we think may have biomedical reasons, and that was the reason for my disclosure. And Jianshu Li, who did the work with the hagfish, and others whose work I didn't mention so much. And I would like to close with thanks to the National Institutes of Health, who've supported my work over the many years that I've been working. And the not mentioned here, Howard Hughes Medical Institute, without whose support we never would have started these fishing expeditions. And also the Emory Vaccine Center and Georgia Research Alliance in particular for helping us set up our laboratory at Emory University. And finally, thank all of you for listening to this long, fishy tale. Thank you.
You'll answer some questions. Yes. All right, I would like to thank Dr. Cooper for such a great and enlightening talk this afternoon and open up the discussion to questions people might have on this presentation. All right, maybe I'll ask one. I was struck by some of the cytokines that we see in the lamprey, and I'm just curious as to your thoughts as to why are some kept or maybe some not kept, or some that originate so early, such as IL-17 perhaps, and some of the others, and also BCL6. Why is BCL6 in the B cell but perhaps not in the T cell origin?
I wish I could answer all of those questions. Interleukin-17, as you mentioned, is a very ancient cytokine, although it's been relatively late in its discovery by immunologists. Even oysters have what is thought to be an ortholog of interleukin-17. What's surprising to us is that the ones that we've not been able to find, and in particular interferons, they play such an important role in our defense systems that we were sure we would see those and their receptors. There are many of the genes that are interferon responsive genes, and something must be activating them. We have found one interferon receptor gene, the interferon 1 receptor gene, look-alike. So it's possible that we've just missed it so far. The genome sequencing is not perfect. It's also possible that something that structurally serves the same purpose that wouldn't be identified by nucleotide sequence similarity is also present. I won't go on.
So in the sea living animals, are there lots of viruses and bacteria like in the atmospheric system we face every day? Why they are making all these different adaptive immune systems?
I can't answer that question, but it seems striking to us that every jawed vertebrate that has survived has one or the other of these systems. It could be that they have been key to selective survival of lampreys and hagfish and the jawed vertebrates that we know about. We don't know a lot about their pathogens. For lampreys, there have been outbreaks that have caused widespread death among lampreys in captivity, bacterial in origin. We don't know much about viruses at all, but all of them, both of them are swimming in a sea of bacteria, of course, and there are also viruses in their environment. And as the CRISPR-Cas system, for example, is a way that bacteria defend themselves from plasmids and viruses. So one would expect that these jawless vertebrates would have encountered viruses, potential pathogens as well. But we're mainly just, as for many of the features of this immune system and its protective value, or have so little information that it's mostly hand-waving.
Do they also have myeloid systems similar to the mammalian system? You mentioned extensively about the lymphoid systems.
Oh, they do have. They have all of the different types of blood cells that we have, essentially. They have granulocytes, some of which look like eosinophils, others of which look like basophils. They have monocytes and nucleated thrombocytes, which serve as blood clotting cells, red blood cells, of course, which are nucleated. Basically, they have the whole repertoire. There's been a recent publication that shows or provides evidence that endothelial cells in the blood vessels are a vertebrate invention and are not seen in tunicates, the sister group, nor in the Amphioxus, the head of the chordates. And moreover, in the Amphioxus, although they have a heart that's pumping and a vascular system, the reports say that the blood is acellular, that there are no cells floating around in it. But when those evolve, the whole hematopoietic system, hematopoietic stem cells, we're still left guessing at this point.
Thank you very much for a beautiful talk. I'm just curious, is there any relation between these VLR gene products and complement system? Some interaction or maybe some mechanism to combat bacterial infection or something like that?
There is a Professor Lee and his colleagues in Dalian have published studies in which they showed that VLRB antibodies coupled with C3 and a C1q ortholog from lampreys together can induce lysis of bacteria and tumor cells. So at least in vitro, these can engage complement components, these studies suggest, for a lytic protective, potentially protective activity.
And just a very short question. Do you think that similar to EGM, there is a change of conformation from planar to staple form or something, because it's very similar to these antibodies in a human.
We assume that the constant region, like the constant region, or the stalk region, like our heavy chain constant region, may also have a receptor to allow them to facilitate phagocytosis, ADCC activity. But so far, that's an assumption without any supportive data.
Thank you very much.
All right. Thank you, Dr. Cooper. Thank you, everyone, for your attendance. And there's a reception in the library, NIH library, if people want to join. Thank you.